Welcome to this educational exercise called Breaking Down the Complexity of Cancer. The goal of this video is to help you understand the cancer diagnosis process, the differences between cancers, and why the cure is taking so long. Imagine that you've had this weird sickness, or maybe a mole that looks asymmetrical, so you decide to visit a doctor. If something seems suspicious, they might suggest getting some scans. The scans help to locate the potential tumor if it's inside of you. However, you'll still need to get a biopsy done. The biopsy involves taking a small sample of the area that is potentially cancerous. This sample will be taken to someone called a pathologist. The pathologist will analyze it to figure out if it is cancer, and if so, what type. From now on, you get to be a pathologist, but to do that, we have to take a closer look. Cells are the units of all living organisms that keep us alive. Groups of cells in our body are called tissues. Normal cells consist of a small information center called the nucleus, which contains our information strings called DNA. There is also a lot of fluid in the cell called the cytoplasm. The nucleus and other parts in the cell float around in the cytoplasm. To see these different parts of the cell, the pathologist stains the tissue on the microscope slide. After staining, the nucleus will look dark purple, and the cytoplasm will look pink under the microscope. Cancerous tissue has fast-dividing cells, so the nucleus looks bigger and the cytoplasm looks smaller. This is because there is no time to make cytoplasm. Another feature you'll notice is that the cancer tissue will look like blobs. The cells have no clear patterns. They are no longer following our body's boundaries and can create tumors. Now that you know what cancer looks like, let's try applying it with a question. Which colorectal tissue is cancerous? Colorectal tissue is found in our colon and rectum, which are part of the large intestine. Go ahead and take a guess. A or B? You can pause the video here and click play when you're ready to hear the answer. The answer is B. If you take a look at A, you'll notice that it is very well differentiated. This means that you can see the differences in the different tissues. The top layer, the middle layer, and the bottom layer look different, and they are in nice, organized patterns. On the other hand, B is poorly differentiated. The borders of the tissues are mixing into each other, making a blob. If you remember from earlier, each cell has a dark purple DNA center called a nucleus, and lighter liquid surroundings called the cytoplasm. You can see it clearly here. Cancer cells have bigger nuclei and less cytoplasm because they divide too fast. Alright, let's have a brief on grading. Grading tells us exactly how messy the cells are. What do these cells look like under the microscope? Well, if it's a low grade, the cells are well differentiated, and the normal patterns in different cell types can be seen, with fewer dividing cells. High-grade cancer, on the other hand, means that the cells are poorly differentiated or undifferentiated. They all look the same and the tissues are mixing into each other. It looks like a mess. The other feature is that there are many dividing cells. With that, let's try another question. Which one is the high-grade bladder cancer? A or B? You can pause the video and hit play when you think you've got it. The answer to this question is B. Taking a close look at the pictures, you can see that A has some dividing cells, which look like stretched circles. It also has different visible tissues with a distinguishable pattern. B, on the other hand, has more dividing cells and a mass of one type of tissue. But don't forget, A and B are still cancerous. A normal bladder tissue looks like this one over here. You can see clear patterns and layers of tissues that look very different. Now let's do a brief on staging, which tells us how much the cancer has spread. The exact stage ranges that are used are specific to each cancer. The typical numbering system is from 1 to 4. Low-stage cancer refers to a tumor that is easily removable because there is no invasion into deep layers of tissue or into blood and lymph vessels. In the picture below, our little friend is digging their hole not too far in the ground, so it doesn't hit the dangerous red zone, which represents our blood and lymph vessels. High-stage patients will have metastasis of the primary cancer. This means that the original cancer has invaded into other tissues and the blood and lymph vessels, which carry cancer cells to other places in the body. The higher this stage, the more dangerous it is for the body, because it is harder to find and treat the cancers. In this picture, our friend has dug their hole too deep, going into the red zone. The hole has reached the high stage level, and our friend will get into some trouble. Alright, let's try out a question. Left or right, which is a low-stage melanoma? Melanoma is a type of skin cancer. You can pause the video to think. When ready, click play. The correct answer is left. You can see that on the left side, the melanoma has not really invaded into the deep tissues yet, whereas on the right side, it has gone all the way down into the deep tissues of the skin and into the blood and lymph vessels. This can become dangerous because it allows for metastasis. The final factor that you need to know as a pathologist is tissue location. There are two types when talking about cancer. The first is a primary location of the cancer, which is where the tumor started. The other one is secondary location, used when the cancer spreads. This is the metastasis that we talked about. The place where the primary cancer migrated to is the secondary location. In the picture below, we can see that the primary cancer is in the intestines, and it metastasized to the lung and liver, which are the secondary locations. However, one should note that the cancer in the secondary locations are still intestinal cancer cells. So, we still use the treatment for the primary cancer, which is intestine cancer, in the secondary locations as well. To help you understand what I just said, let me explain what makes one cancer different from another. You've seen on microscope slides that cells look different based on where they are, and this is because each group of cells have different functions. For example, the cells in your brain send electrical signals to your body, and the cells in your skin protect your insides from the outside. Proteins are the things that allow cells to look and work differently because they are the cell workers that do everything for the cell. Different proteins means different cell types. Cells become cancerous due to different problems in the information strings of our cells, called DNA. Our DNA makes our proteins, so bad DNA means bad proteins. Together, the different proteins of each cell type and the different problems in the DNA make different cancers. Okay, so I said that we can't just tell what kind of cancer is present just by looking at where it is in the body. So how do we know the cancer type? Well, I just told you that different cell types have different proteins, and that cancers have various bad proteins. So if we look for these specific bad proteins called biomarkers, we should be able to trace it back to what kind of cancer it is. Here we have the two different biomarkers on these two tissues. To figure out which is which, the pathologist stains the two tissues. Here we go! We found that the one that picked up our green stain is stomach cancer, with the biomarker called CEA. The one on the right picked up the blue stain. This one is breast cancer, with the biomarker called ER. Let's do a review of this information for the next question, which is stomach cancer versus breast cancer. Generally, the stomach has muscle tissue to move around our food, and glandular tissue to make acid to help break food down. The breast tissue is meant to store fat, so they have fat or adipose cells. The female breasts also have glands that make milk for babies. Below is a picture of what the tissue organization for the stomach and breast looks like. But like we said, to tell what type of primary cancer we have to use biomarkers. The common biomarker for advanced stage stomach cancer is CEA. A common biomarker for breast cancer is ER. There are other biomarkers too, because there can be different proteins that went bad in each cancer. Okay, last question. Which one is a stomach cancer? A or B? Pause now to think if you want. The answer is A. Stomach is made of muscle and gland tissues. It looks similar to the normal stomach picture you saw earlier. Breast, on the other hand, has fat, skin, and gland tissues. But the real thing that tells you which is stomach cancer are the two biomarkers. A has a CEA biomarker, which is for stomach cancer, and B has the ER biomarker, which is for breast cancer. As a refresher, do you see the cells labeled tumor having more nucleus and less cytoplasm? That's the cancer, but the type of cells and the places in the body are different. Great job with the pathologist work. You can tell that the job is important because all of the factors you've learned today play a part in deciding treatment for the cancer. We've talked about proteins, location, staging, and grading, but there are also other factors that we haven't mentioned. We know that all of these factors come together to make each cancer different, so that means each one must be treated differently too. Let's consider each treatment to be like a tool in our cancer toolbox to help you understand the process of curing cancer. We treat each cancer using this cancer toolbox. Researchers make new tools for our toolbox. Doctors then use these tools to treat people with cancer. After that, doctors tell researchers about what happened and what new tools are needed to treat more patients so more people can survive. Each cancer type needs a different tool. This process of making and testing them takes a lot of time and money. Thanks to people like you who are willing to understand cancer and support the scientific process to grow and better our cancer toolbox, we will be able to save more lives each day. So thank you.